A very good evening to everybody, and welcome to another episode of The Different News. Today, I have a very interesting scientist and my guest here, Dr. Krishnamohan Atmakuri, with me here. So, Dr. Krish is Associate Professor at Translational Health Science and Technology Institute, India, and he is aggressively working in the field of infectious diseases. Welcome, Krish, and thank you so much for accepting my invite for coming on my podcast. And I think we have a lot of things to learn from you today.
Thank you, Dr. Gargi. It's wonderful to be here, and thank you for the opportunity to share my thoughts and some awareness on the infectious diseases with your audience.
So, Krish, first of all, I will ask you my favorite question. That would be about your journey, because as much I know about you, you have a very extensive career in terms of you started with biotech. You move. I think you haven't left any part of biology to touch upon that way, like immunology, and then you are working in the field of infectious diseases, and particularly the thing today we came up here is to talk about sepsis. So, before going into all those things, it will be very interesting to know this versatile kind of career that you have had, and still you are ongoing on it. Would you like to share your journey with us?
Sure. So, I think as a kid, I was always fascinated with medicine, and somehow I was also fascinated with diseases. I don't know why, but and to some extent the microorganisms. I think the stimulation was me looking at a microscope when I was possibly 10 years, trying to pull out different materials from the soil, leaf, and flowers, and just keep looking at what are the kind of small things that we can see. And I think that fascination grew as I started learning biology. So I think I would have scored some of my maximum scores in biology all through my schooling and the college. And so, of course, I was more fascinated to become a physician and a cardiologist, but cardiac surgeon, to be actually honest about. But at that time, the medical colleges were less and the competition was very high. Didn't get much of an opportunity to be in the area of that I wanted. So I said, I'm not going to get into any other area. And since I was not that much interested in engineering, went into doing my bachelor's in agriculture, taking the plant sciences as an opportunity to get into the biology. And then I did my master's in plant biochemistry, wherein we had a lot of learning a lot of interesting aspects of biochemistry in general and a little bit specific on the plants. But also we had a couple of courses on humans and mammals in terms of the biochemistry. So that led me to get into the PhD, where I went to one of the pioneer plant molecular biologists in the country. And I told him I don't want to work on the plants, but I wanted to work on the plant pathogen. So there's a plant pathogen called Agrobacterium tumefaciens. So now it is very well known as a plant genetic engineer. At that time, it was emerging to be a plant genetic engineer. And there was a lot of excitement in terms of understanding the biology of the Agrobacterium. So Agrobacterium, interestingly, delivers a portion of its DNA into the plant cell, called as the tumor-inducing principle. So it derives the two hormones which are important to distinguish and differentiate a plant cell into their shoot or a root. And then it forms tumors at what is called as the crown region, which is the intersection between the root and the shoot system. And these crown galls can become sort of mini factories, if you will, releasing some of the carbon and nitrogen sources, which are highly specific for Agrobacterium. So Agrobacterium continues to be in the soil, in the rhizosphere region of the soil, and is able to manipulate the entire plant system towards its needs. It's a fascinating biology, and I wanted to be part of understanding the biology at the molecular level. And that paved way for me to get into doing my first postdoc in the Texas Medical Center, in again one of the pioneers in the type 4 secretion system. It turns out that Agrobacterium utilizes a particular molecular machine which is made of 12 different proteins and 3 or 4 accessory proteins, and delivers this DNA as well as the proteins through that particular channel. So in that particular postdoc opportunity, we did understand some of the molecular mechanistics of this particular machine. And then I moved on to work a little bit on tuberculosis in Harvard Medical School, rather Harvard School of Public Health in the Department of Infectious Diseases and Immunology. Again, with one of the pioneers now in tuberculosis research, wherein we asked the question, is this molecular machine present in MTB? It turns out that the molecular machine of Agrobacterium is sort of conserved to a large extent in Mycobacterium. So the question was that whether the kind of functions that the Agrobacterium was doing, would the Mycobacterium machine also do similarly? So to some extent, thankfully, I think we didn't see the DNA translocation in the Mycobacterium tuberculosis, because that would have been really worrisome, that a pathogen DNA is getting into the human cells. That would have been really worrisome. But it turns out that it is able to still deliver some of the pathogenic proteins which are used for different modulations of the host cellular processes. It's not that we were the ones we discovered that. There was quite a bit of literature by other groups doing this, but we were trying to understand different aspects of how this particular machine, loss of this machine, would make changes globally on this particular pathogen surface. So that is how I started my journey in terms of moving from the plant science through a particular plant pathogen all the way to a human pathogen. And it to make it much more complex, after I joined THSTI, so ended up getting an opportunity to be part of a lecture which was delivered by one of the very distinguished professors from AIIMS, New Delhi, on sepsis. And that basically led me to understand the sepsis, sort of neonatal sepsis, especially being a black box, and wanted to contribute to see whether how we can make this better and understand neonatal sepsis better. That's how my journey has come to current state.
Thank you, Krish, for, I know it's a very long journey, but you summarized in only few minutes. It is so interesting and amazing. I mean, how you shifted. I mean, it looks like all the dots were there, and you were the person who were supposed to connect it, hopping on from one point to the other.
Correct.
It is interesting. So nice.
Thank you.
So thank you, Krish. So, Krish, you mentioned about sepsis, particularly the field that you are into, sepsis in neonates. As we know that it is a critical global health challenge that way. So, what are the key barriers of early detection of this? Are there any possibilities that we can actually detect it early? And if so, what problems or challenges we usually face? If you can enlighten us, it will be really nice.
Sure. So I'll just give a brief outline of how the sepsis works and what are the challenges in this area, and then go into the clinical side of the diagnostic challenges and how they do it. So it turns out that sepsis for a long time was considered as, by some people, some groups, as triggered by infections. And some groups basically thought it was more of an autoimmune disease. Because there are a lot of cases where you actually cannot find out what is the source of that infection, what is the pathogen which probably is triggering. And because of lack of that information, so individuals thought that it was primarily because of autoimmunity. And so But in 2016, a group of distinguished clinical um, uh, distinguished clinical teams basically assembled in Europe, and then came up with a definition which sort of is that sepsis is caused because of a dysregulated host response to an underlying infection, which can lead to multiple organ damage and possibly leading to death if not controlled. So, um, sepsis in neonates is more challenging because a lot of preterm neonates who weigh approximately 1500 grams and are between 28 to 36 weeks. So different groups classify them as preterm depending upon the literature. If you see 28 to 34 weeks, 28 to 36 weeks. But in general, these are the most vulnerable neonates. And of course, in terms of adults, anybody who is immunocompromised can be susceptible to sepsis in the sense that, infections happen all the time. It's just that some of the times the infection basically goes uncontrolled. It's like using a sledgehammer to kill an ant. So when you try to over bear on the infection and control it, then the challenges can happen, and that is when the dysregulation happens. And in neonates, possibly in the preterms, there is a lot of immatured immune responses and host responses, and that's the reason the pathogen is able to overwhelm very quickly. And there is a subclassification in the neonates in terms of when they get infection as early onset sepsis, which is thought to be that it is coming from primarily the mother, because it happens within the first 3 days after birth. And anything beyond 3 days is considered to be possibly coming from the hospital acquired infections, or it could be coming from people who are handling the baby, who could be other than the mother. And so there could be potentially hospital reservoirs, there could be potentially reservoirs because of people coming into the hospital. And so the chances of the neonates coming in contact with these pathogens is tremendous. And so the challenges are in terms of, so just to give you some numbers in terms of how the burden is. So WHO basically in 2020-21, and also the recent Sepsis Congress in 24, came up with these numbers where they said approximately close to 50 to 55 million individuals in the world every year are suffering from sepsis. And approximately 10 to 15 million individuals die because of sepsis. And so it's a big, big challenge across the globe. But interestingly, 70 to 85% of this burden is primarily within the low and middle income countries. And so it is quite a challenge in terms of making sure that this burden is reduced. I think one of the major challenges in LMICs is in terms of the diagnosis and the early diagnosis. It's not only the diagnosis, it's the accurate diagnosis, the cheap diagnosis. There are some tools available which are still yet to come into some of these LMICs because of the cost issues and the supply and the demand issues. It becomes challenging. And so most of the LMICs still follow the blood culturing, wherein an ml of blood is basically taken from the patient and cultured in artificial media in order to make sure that you can first detect the pathogen and then try to see what that pathogen is. And in terms of the susceptibility of this pathogen to some of the antimicrobials, because what is most important is not just identifying the pathogen, you should be able to quickly also determine what is the kind of therapeutic strategy one can utilize in order to control this infection. And in neonates, because of the poor or immatured immunity process, so it is quite possible that the pathogen is able to quickly overwhelm. And so the blood culturing can give you the data anywhere between 24 to 96 or even sometimes 5 days, 6 days. It all depends upon what is the blood, the pathogen load in the blood. Sometimes the pathogen load is quite less, could be anywhere from 1 to 100 bacteria per ml. So if in neonates you cannot even collect 100 microliters or 200 microliters of blood at any point of time from these neonates who are just born, so it's quite possible that you may not be able to detect the pathogen. So a lot of cases go as defined as blood culture negative, but they continue to show clinical uh, symptoms of sepsis. So the diagnosis is one of the major criteria. If you talk to any of these critical care neonatologists, pediatricians, they always say that diagnosis is one of the most important things. Then, of course, the diagnosis has to be done as quickly as possible, and then you give the administration of the antibiotics. But because of these challenges, so the clinicians cannot wait. So they have to give a broad spectrum antibiotic to begin with. And sometimes the broad spectrum antibiotic has to be changed depending upon how the symptoms alleviate. And the neonates are always monitored very carefully by the critical care physicians. But it's quite possible that sometimes the neonates are coming not born in a particular hospital. They're basically discharged, they're fine. Within no time, they basically carry on getting infections. They get treated in some other hospital, then they come to the tertiary hospital. They may not have the pathogen at all. Still, they will be showing all symptoms of the sepsis. It is quite possible still the pathogen could be something that can be cultured, but because the pathogen is no more visible in the blood culture, it becomes much more challenging to the clinicians because they don't know whether this is caused by virus, bacteria, or fungi. Right? And so the early diagnosis and the determination of what is the right antibiotic combination that can prevent the sepsis to derail and make it very difficult for the neonate is one of the major challenges. The other challenge is, of course, in terms of the treatment, because you need to give that appropriate treatment. So it's much easier to give that in adults, but in neonates, as I said, things can so change dramatically within a couple of hours. So it becomes very difficult to handle the neonates and control the infections. So primarily the infections are, especially in some of these LMICs, are caused by three different gram-negative bacteria and a few gram-positive. Some of the prominent pathogens which cause neonatal sepsis are the Acinetobacter pneumoniae, the Klebsiella pneumoniae, and E. coli, the pathogenic E. coli. Of course, there is E. coli everywhere, but pathogenic E. coli. So I think there is a lot of literature on in terms of the reservoirs of these pathogens, not only in the hospital settings, outside the hospital settings, but in terms of how one has to, keep the hospital clean and safe is another challenge which can, to a large extent, prevent the burden. So, but in adults, there are other challenges because adults, they get into ICUs because of some surgeries, and they, while they're recuperating from the surgeries, then there is a possibility of infections again. Could be because of hospital acquired, or because they're now to some extent immunocompromised, depending upon what kind of a surgery that is. And so there is a possibility still for them to again acquire infections. And if they are on some steroids, then the challenge becomes that the infection can take over within a very short period of time. Again, their early diagnosis, even before the symptoms kick in, is one of the major challenges. To some extent, I think the clinicians and the critical care physicians have been able to figure out what is the kind of sepsis management, but to a large extent, it all depends upon how early can you diagnose the sepsis.
Yeah, I think that is very critical, the early diagnosis, because as you rightly said, that when it is sepsis, we really don't have time. We
Hmm.
don't have time. And from clinical perspective, again, getting that the correct information in terms of diagnosis, only then, the treatment would be pinpointed, and then the problem could be solved. So there's a lot of
And
challenges in
I wanted
this,
to tell
as
you
I
one more thing, which I missed, which is basically where the when the pathogen kicks in, when you
huh,
need the immune system to respond, the host has to respond to the pathogen presence. That is when the pathogen is also able to subjugate the host cellular processes. So
right.
there is a tug of war between these two, the host and the pathogen. It's the load of the pathogen which probably determines to what extent it can dominate over the host. And you have this seesaw kind of yin and yang sort of a response by the host and the pathogen, wherein sometimes there is a very high immune response, sometimes there is a very low response, there's a septic shock. And also people who recover from sepsis, they have a sequel which can continue all the way from 2 to 10 years. So some people lose their limbs, hands. So it becomes more challenging. It's not like you just, you recover from a flu and just walk off and start your work. So
It's
things can
very
be quite
surprising.
challenging.
It's
Yeah,
very
things can be
surprising
quite
what you
challenging because there could be some sort of internal damages which would have happened to your tissue and your vascular
said.
function. So things can go really bad. So it's very important that it is diagnosed and treated very early on.
Right. Thank you, Krish, for that extensive and comprehensive way of explaining such a critical science concept, and of course, pertaining to the diagnosis and how we can manage sepsis. So, what I understand is that there are a lot of points right now, like few minutes back, you were talking, which even I am not aware about it. So I personally feel that awareness about sepsis, and I think you are quite vocal about it, and you are advocate of that. So my question is, do you think that researchers or healthcare professionals can also have some role apart from scientists like you who are into the core science, working day in and day out to solve this problem? But apart from that, do healthcare professionals or researchers have some role to play in creating awareness about this area? Because I think this is very important, and I'm not sure that much information we hear about it unless, I mean, it's maybe inside your lab everybody talks about the same thing, but outside how much, that's a question. So what would be your advice or suggestions in this aspect?
So, a very important question, pertinent, because I think the awareness is going to be something very important in order to reduce the burden to a large extent. For example, in the last year, so every year, September 13th is celebrated as sort of a sepsis awareness day, wherein a lot of agencies, basically hospitals, then professional agencies, government agencies, basically try to create awareness among the general public. And with the emergence of the social media, it becomes very easy to access the population in a very, very short period of time. I think generating some very small 2 minute to 5 minute videos in terms of the awareness and in terms of what kind of pathogens linger on, what kind of the symptoms can be possible. Because the challenge here is because symptom normally is not anything different from a regular infection, regular fever, and so on and so forth. There are few characteristic symptoms which are a little classic of sepsis, but they basically come up when the infection takes over. So it's a sort of not very early on. And so a lot of the diagnosis happens in the terms of the sepsis is more of a rule out sort of a diagnosis as compared to the rule in. And so it's important for the healthcare professionals and the public health individuals to create that awareness to say when you can sort of suspect sepsis and how early can you bring in somebody who is suffering for quite some time with respect to some of these symptoms. It's not that one has to have all the symptoms because, as I said, different pathogens can cause this. And so you won't have the same symptoms depending upon what pathogen, and also depending upon the pathogen load. It's a question of somebody who is just having a regular fever and suddenly goes into this kind of a situation. And so that awareness has to be brought in. So possibly through enactments of small videos in terms of awareness, and possibly these should be not just played on this annual sepsis awareness days, but all through the year, and possibly at all hospitals, and some of the government health agencies, some of the research institutions like us, because there's a lot of people working in institutions like ours. And so it is important that even those who are within the institute are able to get that awareness and pass it on. So these, I think, is required. In hospitals, of course, you can have banners, posters, and some of these kind of tools which can still be able to spread this information. But in terms of the public health, the professionals, I think it is important, especially in LMICs, it's quite challenging to keep the hospital environment cleansed all the time, given the number of patients coming in per hospital. And that is yet another challenge, how you can keep the hospital clean, how to create that awareness to make sure that people understand the importance of the cleanliness. And there is a disinfection process which happens in hospitals all the time. It's just that that has to be monitored much more carefully. Sampling of the samples from different potential reservoirs in the hospitals can also be taken care of, so as to always be aware of where would the potential reservoirs could be lingering on and how to eliminate them. And so in terms of the transmission, in terms of the awareness to the so-called people who are accompanying the patients and who are bringing the patients, because patients are with particular pathogens, could be having some or the other infections, if at all they're coming because of infectious disease, and so they could leave traces of these pathogens in the hospital. So it's quite challenging and required for the public health to create that awareness. In terms of those who are physicians, I think nowadays there's a lot of population which is educated, and so it is important to have this in local languages, possibly the banners, videos. And if somebody is having some kind of an infection which probably is lingering for quite some time, it's important for the physicians also to share that sort of awareness with the patient relatives and accompanying families, so as to make sure that they understand what can be the problem if it potentially slips into sepsis. And I think that this awareness could bring in a lot of change in terms of how this is supposed to be done. Because, as I said, the rule out should be sort of made into a rule in situation, so as to make things better.
I think these are very pertinent points that you have mentioned, because I think and these are very simple things. It doesn't require too much of paparazzi kind of thing to actually do these things. Like the best thing that I liked you said about creating using social media in the right way, following the correct and ethical guidelines, and spreading awareness about this critical area of medicine and health.
Yeah.
uh, Secondly, even the small places where like you said, in regional language, the pamphlets, like a offline kind of materials that could be there. I think this is very effective, and it's not something which requires a lot of budget or lot of efforts. It's something very simple. So thank you, Krish,
Even in
for that.
general, even in general with the media, right? The TV media,
Yeah,
and so
correct.
they have a huge opportunity to create awareness to the general public. And
Yeah,
so
absolutely.
rather than creating this awareness only on a particular day of the
Yeah,
year, if this can be done on a sort of a health program, sort of a thing which
exactly.
possibly could happen every week, this could really create a lot of awareness.
Yeah, I think it's more of a cumulative kind of effort
Correct.
that is required from various disciplines, in a sense, like not disciplines but various sectors, like scientists like you, other junior researchers maybe who are aspiring in this field, and of course healthcare professionals,
Yeah.
and related people, public health specialists. So I think it really needs to, we really need to take a call on this, and think something about not doing only annually but doing all through the year something. Thank you, Krish, for that wonderful insights that you shared. Well, Krish, we have learned so much in this small time from you about the challenges and how we can actually help people understand these aspects. But let me just get into a little bit into the core of the kind of work that you do. And you have this expertise in bacterial pathogenesis, microbiology, and uh, molecular biology, of course. So as a scientist, how do you see advancements in this field in terms of solving the challenges, maybe for better diagnostics or early diagnosis, or if I put through mm, like even treatments, treatment protocols. So I think your insights are very valuable, so can you please enlighten us on
Sure,
this?
sure. So, as I said, classically for a long time, the blood culturing has been the key tool for physicians. Because when I talk to some of the physicians, they say that sometimes the nucleic acid based amplification techniques may not be very appropriate because sometimes you can have the pathogen killed, but the genomic remnants could continue to be there for some time. So they prefer the blood culturing because if you have a viable pathogen, growing pathogen, then it would be the one which would start culturing. So while that is important, which can continue to be sort of a gold standard, it's important to also understand that, for example, some sort of a DNA chip, some sort of a multiplex PCR, all these could really help to quickly identify the pathogen, even if it is in very small numbers, which might take some time for the culturing to happen. It's also possible that there are a lot of anaerobic organisms which may not prefer to grow in these kind of artificial culture medias. There are a lot of fungi, there are a lot of, to some extent, viruses, which we may not know what is the kind of exact conditions under which they can be cultured easily. So it is going to be challenging if one has to think only in terms of culture. That's why nucleic acid-based detection systems are present, and it's except that some of these tools can be expensive, which cannot be affordable to quite a few people, in especially the LMICs. That's some of the one of the things that could be done quickly to make it easy. But the thing is, detection of the pathogen is not sufficient. As I said, some of these bugs that we also have in our lab are highly resistant to a lot of antibiotics. For example, we have these isolates of Acinetobacter, Klebsiella, and E. coli, which are resistant to 15 to 20 different antibiotics. And so they come with a alma mater, and one has to know how to deal with that. So it might be, you can't just basically say, okay, I'm going to give a broad spectrum antibiotic. Sometimes it might work, sometimes coincidentally it may not work. And so a quick detection and analysis of what are the sort of antibiotics that the pathogen is susceptible to, what is not susceptible to, and a combination of this therapy is what is being tried in quite a few places. And to some extent, that has significantly reduced the burden. But this again is to be done as quickly as possible. Sometimes, as I said, even sometimes if coincidentally you give a broad spectrum antibiotic, it is able to kill, but it is quite possible that because the pathogen was overwhelming, it's not like you kill the pathogen, the symptoms will go away. Because the pathogen always is manipulating the environment. It is releasing some of its molecules, proteins and other molecules. And these can interact with the host molecules and also lead to this the subjugation of the cellular process, which leads to establishment of the infection and the sepsis to kick in. So one has to think about some of these molecular um, kind of a battery of arsenal, if you will, that the pathogen utilizes, and think about how to control them, how to target them with small molecules. So some of the studies across the world are being looked at in that particular area. That's another way to see whether you can deal with sepsis at the molecular level differently. Now, one can also think about, given that there are several pathogens which potentially can cause sepsis. So now, if you were to look at a panel of a pathogen signatures, now it might be that you might get an answer no in some cases where none of these pathogens which are on the panel could be the ones which are causing sepsis. So one could miss out the others. And so now literature is emerging wherein people are looking at host signatures, which can distinguish whether this is bacterial, this is viral, this is fungal. And if we can, so we have some work that we are trying to establish, wherein we want to see whether some of these signatures can indicate sepsis even before the clinical symptoms kick in, because that is the huge advantage that you can get out of this molecular analysis. And so the earliest molecular diagnostics is a real important thing. In addition to that, now clinicians over several years, decades, have collected information about these sepsis patients in terms of clinical data, in terms of the symptoms, in terms of how they develop sepsis, how they basically progress through sepsis, how they recover. So there's a huge body of literature out there. So now with emergence of AI and ML, so one can basically put all these together, come up with an algorithm which in combination with your therapy can basically help control sepsis possibly. So the AI is certainly going to take over in terms of predicting sepsis. In fact, there are a lot of molecular changes which happen during sepsis. The first thing is in terms of some of the immune response cells, which are basically required for taking up the pathogen, antigen presentation cells, some of the cells which can kill these pathogens. There are a lot of molecular changes happening. So some of the groups and some companies are also out there looking at the biophysics, the structure of these cells. Can you predict just by looking at the biophysics of a bunch of cells, so that you can say, yes, this person is progressing to sepsis? And so,
Amazing.
yeah, so there are several groups which are looking at, because you basically have sepsis, can your sweat release some molecules which can be captured in your sweat? The body releases the molecules into the sweat that can be captured, and by use of sophisticated mass spectrometry, can you detect these molecules? So a lot of molecular level detection systems are emerging, and it's a question of how many of these reach from where the technology is developed to the LMICs and the labs. And it's also a question of how much these hospitals are able to afford, the patients who are coming into these hospitals are able to afford, and in terms of how many of these patients have support from either financially from the government or financially from the private insurance players, medical insurance players. So all these will make some challenges, but the emergence of different approaches in terms of detection of the infections very early on in the progression of the sepsis. There are a lot of literature on scientific literature on shortlisting biomarkers. There are two classical biomarkers which have been already in practice in hospitals. Sometimes some hospitals use one of these biomarkers, some of them use another biomarker. There's a lot of studies asking, are these biomarkers good enough? So different studies have shown that sometimes they're good, sometimes they're not good. Sometimes these biomarkers cannot be used for prediction, but they can be used for stopping the treatment. So a lot of development in terms of the changes of how you're looking at the data. But I'm confident that possibly in another 5 years, 10 years, there would be a framework of not just the biomarkers in terms of the molecular level, but algorithms together should be able to predict sepsis very early on. I
So
also wanted to mention, sorry, I forgot, but I
yeah,
wanted to mention, so with the emergence of the omics, the transcriptomics, the proteomics, the metabolomics, right? And with all this emergence of omics, which can now look at globally your response to a single infection, and a combination of these, if you were to look at even very some small subset of molecules, might also throw some light into early detection. Sorry, I missed that point. I should have.
no, I think it's a very promising time. I mean, it's just like, as you said, in the next 5 or 10 years, I think, I mean, it's a very good time with such advancements. Like you are saying, the kind of research or kind of advancements are going on in this field, studying our cell structure. I mean, in terms of biophysics, and then predicting about sepsis or early detection through, some molecules that are spread through sweat. I think these are fantastic and phenomenal advancements. And of course, with science and with translation of science, it's always a matter of time. And thank you, Krish, for enlightening or bringing this up, because I think most of us, though we are from healthcare background or from science background, many of us are actually not aware much about these scientific innovations, even because I think now we have such a good opportunity for different kinds of science innovations and entrepreneurship particularly. So I think many young entrepreneurs might get good ideas out of this interview, and they might be, very valuable asset to the society, and particularly in terms of early detection of sepsis at at several levels. So it is
And,
fantastic.
and if somebody progresses through septic shock and you basically
Yeah.
accumulate a large amount of chemokines and cytokines and
Mm,
proteins which are not really, which are basically derailing your organs. So
mm.
what there is an expensive approach which is done in only some very affordable hospitals, wherein you move the entire blood through a particular column which cleanses them of all these molecules so as to reduce this load of these molecules, which basically is derailing your organ functions. So that way you can recover the organ functions very quickly to a large extent. So some technologies are emerging in that area as well. So there is technology emerging not only in terms of the diagnosis but also in terms of the treatment, and of
Treatment.
course more and more therapeutic molecules which can target specific pathways of the host which basically are responding to infection, and which are overwhelming. As I said, if you were to use something like a sledgehammer to kill an ant, can you reduce that sledgehammer response to a very small response so that you don't actually kill the host because of the host response?
Amazing, amazing. This is wonderful science. It just brings so much of spark in all of our eyes. Thank you so much, Krish, for that. So, one aspect of any successful translation of any research for the benefit of human society or human health elevation is, of course, collaboration. That is how things work. So I would be really interested to know your insights or your opinions about um, whether or how can global collaborations enhance this part of the science, critical science that we are talking today, that is sepsis, and especially addressing the disparities in healthcare systems, maybe worldwide, if we have to think of the big picture. So, please, Krish, if you can give your valuable insights on this.
So again, very pertinent question. Collaborations are very important these days because there is so much very specific expertise with the different individuals. So until and unless you bring all these individuals together for a collaborative approach towards tackling a particular challenge like sepsis, it becomes really difficult. And for example, we have one neonatal project kind of funded by the government of India. And we have 5 different hospitals in this particular study. There are 7 research institutions in this study. And sorry, there are a lot of investigators from the hospital, investigators from the basic research institutions together involving in this collaborative approach to address some of the questions that are very important to understand the biology of sepsis, to also think about whether we can find some specific biomarkers to diagnose sepsis, also to see whether we can look at the molecular level. For example, I was talking about the specific pathogens which can cause sepsis. There is also yet another interesting concept wherein the gut microbiota, which is supposed to be in a particular place in the gut, for different reasons can get leaked into your bloodstream. And the blood is supposed to take care of that. But if you are slightly immunocompromised because of some medicines or whatever it is, genetic predisposition, that can lead to almost sepsis, because there you have a bunch of different organisms which can now overwhelm, and quite a few of them could become opportunistic pathogens, right? And so in general, a pathogen, the best pathogen is one which is going to keep the host without really knowing that there is a pathogen. That would be the best pathogen. But at times when it realizes that there is an opportunity to expand and explode, then there is a possibility that some of this gut microbiota can also cause damage. And it's not that the gut microbiota of individuals is always having very good, healthy bacteria, but because of the food habits, because of different conditions, different so the genetic composition of these individuals, you can have a slightly altered gut microbiota, which can then overwhelm the system and cause sepsis. So you have a different altogether ball game coming because of the polymicrobial sepsis as compared to the monomicrobial, which possibly could be because of one particular pathogen, right? So now, to understand these, you certainly need that collaborations. As I said, we have this multi-collaboration project. It's a multidisciplinary, multi-hospital program. And I think these kind of approaches are necessary. The government understands it, the funding agencies understand this. And that is where things are going now. And this can be within the country, this can be global. And it all depends upon who has the expertise, who has the interest to look into some of these aspects. Now, we have the technology in this country, we have the expertise in this country to look at all these aspects that to a large extent need not be international. I don't mean that we should not promote the international collaborations, but India has that capability. And so it is important that we should be able to solve a lot of the sepsis burden that is happening in this country.
Absolutely, absolutely.
In this collaboration, I think the physicians are also required. As I said, transmission is one of the major things which is important. So clinicians will certainly come in terms of a better management of the sepsis and the early diagnosis. The diagnosis tools has to be provided by the researchers and to some extent the companies which are making them. And in terms of the insurance companies, in terms of the government basically making this much more subsidized so that it becomes easy for patients to afford some of these particular technologies. Then in terms of the the public health professionals, as I said, they have to think about in terms of not just the awareness, but in terms of how do you reduce that burden and how do you prevent the hospital-based transmissions of these pathogens. And I think that is where it's important that this particular collaboration would be able to solve this. But there has to be a common goal, there has to be a common approach, and one has to think in terms of how quickly can this be done to alleviate the sepsis.
Thank you for your wonderful insights. I think this is amazing, and we got lot of value and lot of learning from only few minutes. Though it feels like very few minutes to me. I think we can talk the whole day, but yeah, we have to draw a line when it comes to a conversation like this. But I think, Krish, the kind of insights we got today, the things of particularly the advancements that is happening in this field, and the role that we need to play as healthcare professionals or researchers, and the multidisciplinary approach, and the strength of India as a scientific community. I think, I mean, there are no clouds, it's only silver lining, which I see now after learning from you. Thank you so much. So, Krish, any last message that you would like to give to everybody from your side? Because we talked a lot about sepsis today and many things we learned from you. So, any last message from your side?
So I want to end with a very interesting small story, a snippet, which actually is very interesting and very much in line with sepsis. So it turns out that after the World War happened, so some of the European countries which were bordering Russia went on to become highly developed countries, while that portion of the Russia continued to be, though Russia is developed, some portions of these borders continued to be villages and farming and agriculture and so on and so forth. So what happened was that some of these children and young adults were developing, to a large extent, infections in the developed countries where there is completely industrialization. While on the farming side and the agriculture side of the Russian population, people were happy. So there was a lot of analysis going into why suddenly, because one has industrialized, why is the infections actually kicking in? Why is that not happening on the Russian side where the farming is going on? So there are a lot of immunologists, infectious disease biologists brought into. It took almost for them to understand after quite some time that the children and people who were working in the farms and coming in contact with the soil all the time. While in the industrialized parts of the world, a lot of children don't even touch the soil, for example, for months together. So one of the recommendations of these bunch of investigators who basically looked at all these details was go and play with the soil at least 15 minutes or 20 minutes a day. The reason it turned out to be that there are some bacteria present in the soil which actually trigger some of the host immune response in these particular individuals, keeping infections at bay. So you need to expose yourself to some extent to sort of these pathogens. I don't mean the pathogens, but in general, the microorganisms are lingering everywhere. So because when COVID happened, everybody was sanitizing left and right, and nobody was touching any surfaces and so on and so forth. So one of my mentors in Harvard used to say, parents want to be microbiologists, children want to be immunologists. So
Wonderful.
yeah, so indicating that how these interplay is very, very important. It's not like you can just sit in one corner of a building in a room which is 100% clean and say that you're going to be always immune to these infections. So it is important that one needs to be exposed to some of these so that they don't become completely immunocompromised. So with that note, I will stop here, and hopefully some of this discussion that we had will stimulate some of the young minds to do more. There's a lot more. I've tried to keep this entire discussion very simple without much of a scientific jargon so that the wider audience will be able to understand this better. This is more of a, I think, an opportunity, thanks to you, to create awareness about sepsis in general, because we did have the ISF 2023, which was in THSTI, and we had these two small dolls which represented the neonates, sort of sized babies with very initial symptoms of sepsis and very developed sepsis. And teachers, parents, children were very enthusiastic, very interested to understand what is it, why is this important. So I think these kind of platforms also will help. Scientific conferences, all these should be helpful to bring awareness into the general public. I have a feeling that if we can create awareness among the general public, to some extent, we should be able to reduce the burden, because sometimes ignorance is considered as bliss. And so people continue to do what they do and might lead to unnecessary exposure to some of these pathogens.
Right. Thank you, Krish. It was wonderful talking to you and learning so much from you. And I think time is very less to gather so much information. But I think, as you rightly said, that you have presented us in such a simple way with stories. And I really love the way you explain things in terms of, I would say that you are a fantastic storyteller
Thank you.
because, yeah, because explaining the science, such critical aspects of this complex area of science and medicine, it's not an easy thing. And I think it was a really fantastic conversation. Thank you so much. Thank you for
Thank
your
you.
time.
Thank you. Thanks for the opportunity, and it's a pleasure talking to you.
Same here.